Well, thank you for the kind introduction. And thank you to the ICP Network Initiative and the organizing committee for the opportunity to present this work. This work was previously presented at the ISMRM annual meeting with the abstract number 1753. So breath holding elevates the CO2 in our body, which induces vasodilatation, increasing the blood that gets into the brain, known as cerebral blood flow. The increase of the cerebral blood flow induced by breath holding is known as cerebrovascular reactivity, which is the normal response of the brain to vasoactive substances. So in brain gliomas, the vascularity is abnormal and is believed to disrupt the perivascular organization, causing expected response to the hypercapnia to be different from normal tissue and heterogeneous within the tumor tissue. So using arterial spin labeling or ASL in MRI as a perfusion quantification technique while doing breath holds, we can directly measure the cerebrovascular reactivity. So the goals of this work were study the feasibility of measuring the CVR using ASL with a breath holding paradigm in patients with high-grade gliomas, evaluate if the CVR response to breath holding is robust enough to see difference in areas within the tumor and adjacent brain tissue, and study the potential use of CVR to differentiate between grade 3 and grade 4 brain gliomas. So in arterial spin labeling, just to give a small summary about the technique, in the brain we have a small labeling plane in the neck in which we label the protons in the water molecules of the inflow in arterial blood. Labeling means changing the blood magnetization. The spin magnetization of the proton to water of the molecules are inverted. The label is obtained by combining radiofrequency pulses and label bearings to reverse the longitudinal magnetization of the water protons in the labeling slab. And after a short period of time, once the inverted blood reaches the brain, the labeled blood of the image is acquired. So the control image does not have the inversion of the proton to water molecules, and the subtraction of the control and the label images result in a proportional perfusion weighted image, which is proportional to tissue perfusion and can be quantified to physiological units with an appropriate mathematical model, resulting in a CVF map. This variant of ASL, or it's called pseudo-continuous ASL or Picasso, and it's the one we use in our research. Are you seeing the slides like flowing? Because I think I'm stuck in the first one.
No, they are moving.
Okay, perfect. So here are some technical aspects of the Picasso sequence we use, as previously described by Villareta et al. With this sequence, we achieve a 3 seconds time resolution doing sync interpolation of control and label images to double time resolution. In a previous research by our group with healthy volunteers, we acquired continuous ASL images while the volunteer was doing breath hold for 21 seconds. In the top row, we can see there is a continuous CVF maps while the patient was doing a breath hold. And there is an actual increase in the CVF a few seconds later, the end of the breath hold. In this research, we saw that 21 seconds breath hold was enough to cause an increase in the CVF, especially in gray matter. So what we did for this project was an adaptation of this respiratory paradigm. We set an ASL acquisition by doing a breathing paradigm of 40 images of baseline normal breathing at the beginning, 120 seconds, followed by 21 seconds of breath holding, interleaved by 42 seconds of normal breathing to help get to the baseline CVF in 10 complete cycles for a total scan time of 12 minutes and 30 seconds. All images were acquired in a 3 Tesla Siemens scanner. So the respiratory trace was monitored by means of respiratory values from the MRI during the image acquisition. A model automatically generated to obtain the actual periods of the breath hold, as you can see here. And this obtained regressor was later modeled as a ramp, as we previously reported higher CVR values with a ramp model, better matching the CVF signal. The first period of the breath holding, the first period of the normal breathing was used to obtain the mean baseline CVF. And there is a delay, a reported delay between this RAM model from the respiratory signal and this CVF signal. So what we did was to do a cross-correlation between the regressor and the CVF to look for the optimal delay between both signals. We tried cross-correlation from 0 to 30 seconds, and the highest cross-correlation coefficient was considered as the optimal delay between the signals. With the optimal delay, we then corrected the RAM regressor to the time resolution of the ASL images, and this delay regressor was used to calculate the CVR using a general linear model. We also calculate the CVR as percentile of signal change. And we also acquired T1 weighted anatomical image, pre and post contrast administration, and a 32 image, and a time of flight image located at the neck to actually see to locate the position of the labeling plane. An experienced neuroradiologist manually draw maps for the tumor and contrast enhanced and non-contrast enhanced areas, and for the edema. These ROIs were later used to do the calculations for baseline CVF and CVR. And after the acquisition, all ASL anatomical T1 on FLAIR images were realigned and quadridistrict to pre-contrast T1 weighted anatomical image. For the statistical methods, the differences in CVF and CVR in all patients were evaluated using an ANOVA or Friedman's after appropriate normality test. And differences in CVF and CVR for grade 3 and grade 4 subgroups by ROIs were evaluated using a t-test or Wilcoxon as appropriate after normality test. And for correlation, we did an experiment rank test for both variables. So here in the images, we can see in the first ones, anatomical T1 pre and post contrast and the FLAIR T2 image. The mean quantification of the CVF and the CVR for these ROIs were used to compare for differences between grade 3 and grade 4. In the top row, we have a grade 3 representative patient with a multifocal lesion, but we only measured the areas depicted in the ROI regions. And in the bottom row, there is a grade 4 representative subject. In the CVF maps, we can see there is an actual increase of the CVF in the location of the tumor, with a marked decrease in the area of the tumor and surrounding area in the CVR maps, as you can see here and here. This is especially marked for grade 4 patients. So there were 8 patients with an histopathological confirmation of grade 3 glioma and 14 patients with grade 4. These patients were classified using the 2016 WHO classification of brain gliomas. All experiments were conducted between May 2021 and May 2022. 7 of the patients were females with a global mean age of 57 years. In this graph, we can appreciate the general mean baseline CVH values obtained for patients in each of the ROIs used, with main difference between contrast enhanced and non-contrast enhanced and contrast enhanced and edema. This for the CVF. And then for the CVR, in this graph, we can appreciate the general mean CVR values measured as percentile of signal change obtained for the patients in each of the ROIs, with main difference between the contrast enhanced tumor mask with the non-contrast enhanced tumor mask and contrast enhanced tumor mask with edema. In this graph, we can see the obtained CVH values of each of the ROIs and the 2 subgroup populations divided with grade 3 and grade 4 using tumor grading. And there were no statistical significant difference between grade 3 and grade 4 in the CVF. Related to this, systematic review by Al-Sayedi found that absolute CVF can differentiate low grade from high grade gliomas, but it loses its discriminative ability when only grade 2 versus grade 3 or grade 3 versus grade 4 gliomas are compared. But if we take a look in this graph, we can see the obtained CVR values measured as percentile of signal change for each of the ROIs and the 2 subgroups of tumor grading. And we can see there is a statistical significant difference between grade 3 and grade 4 in contrast enhanced mask and edema mask for the CVR. In general, CVR values for grade 4 are lower in grade 3 than grade 4, in grade 4 than grade 3. Whole brain CVR impairment has been found in diffuse glioma patients, grade 3 and grade 4, compared to healthy controls by Fiestra using functional ball MRI technique. Our results enhance the greater impact in cerebrovascular activity of grade 4 gliomas in both the tumor and perrelational edema compared to grade 3. And this perfusion method can be helpful to differentiate between glioma grades. Finally, in this graph, we're going to appreciate the correlation between the baseline CVF and the CVR measured in the contrast-enhanced tumor mass. And notice that as the tumor has a higher baseline CVF value, the CVR decreases. So in conclusion, with this study, we demonstrate that ASL with a breath holding paradigm enables the measurement of CVF and CVR in patients with gliomas. CVF and CVR were different across the ROIs used for the patients. There are statistically significant differences in CVR between grade 3 and grade 4 gliomas, even when this difference is not appreciated in the baseline CVF. Of course, more studies are needed to determine the useful of this method to assess brain gliomas. To finish, I just would like to thank the Spanish Ministry of Science and Innovation, Instituto de Salud Carlos III, and AES program grant for grant support, and Fundación Carolina and University of Costa Rica for PhD support. And of course, to all of my colleagues at the Clínica Universidad Navarra in Pamplona, Spain. Thank you.